Okay, I guess I can start the session now. So, good
Uh,
evening. My name is, I can start.
yes, sir, we can start.
Okay, sure. Okay. Good evening. My name is Dr. Shakthi, and we'll be covering cell and general physiology, the second part today, where we'll try to complete your general physiology. So, this is part of a series which is exam mastery live class session for a duration of 30 days in order to help you prepare yourself for the exam. So, this is one way by which you can, you know, understand the questions that come for the exam and do a rapid revision of it and understand what are the high yield topics for each subjects. Okay. So uh, in today's class, we'll be mainly dealing with these set of questions, like what is the cell membrane? Then we'll talk about the apoptosis because that has recently been coming as a question quite often. Then about fixed law, what is simple diffusion, facilitated diffusion, active transport, passive transport. These are all part of your transport across the membrane. So we'll be talking about your membrane transport across the membrane, which includes your facilitated, this simple diffusion, facilitated, active, passive. And then, of course, a very important question, that is sodium potassium ATPase. Okay, so the thing about this topic is uh, because of the new CBME curriculum, apoptosis has been coming a lot. Apart from that, this has always been one topic in general physiology where essays can come. This whole thing can be a very long question. It can come as an essay, part of a sub question, or even short notes for that matter. Okay, so this is a very important topic with regard to your physiology because in general physiology, a lot of questions come from these topics. Okay. So before we start, we'll just briefly guide you through the various subscription plans that are available. There are basic, standard, and mentorship programs. And you can use my code Dr. Shakti20 for 20% discount on subscription. The core features of the app is one, the live sessions, like the one you're attending. There is also pre-recorded sessions of theory, practical, and there is an examination module also for revision purposes. And then there are notes as well as MCQs, which are made available. And for those of you who need a one-on-one help, you can book a slot for personalized counseling session as well. Okay, so we're gonna start with today's topics. So that is your plasma membrane. So what is fluid mosaic model? So the theory behind how your membrane is, you should think of it as a lipid structure. Okay, there is a phospholipid layer which is there, and that phospholipid has one phosphate region and a lipid region. So the phosphate region binds to the water outside and the water inside, and forming a layer of lipid. Now, in the lipid, your proteins generally float across it, creating a mosaic pattern of your lipid with floating proteins, which is called fluid mosaic. Okay, so what is the important question that will come from your cell membrane? So what are the different composition of your membrane? That is a often asked question. Okay, so what you have to understand is if a composition of your cell membrane is asked as a question, you'll have to talk about your proteins, which make up around 55% majority, but you can say that it is almost half. And then we got the lipids, which make up around 40 to 42%. Okay. And then we have your carbohydrates, which make up the remaining percentage. So majorly it is protein and the lipid. In the lipid, we have your phospholipid, which is the major lipid which is involved there. And then we also got the cholesterol. Okay, so this is just a major component. There are still few other varieties of lipids which are there. Okay, so now that you understand the composition of cell membrane, out of that, one thing I want you to understand, this question comes as MCQs. If you look at the protein lipid ratio, so what we saw was, protein was more. Okay, so if you look at a protein lipid ratio, it'll be slightly more than 1, mostly. Okay, so if you look at different components of cells, you'll notice that the myelin has the lowest protein lipid ratio. As in, over there, the lipid is so much more. Okay. On the other hand, if you look at your mitochondria inner membrane or your sarcoplasmic reticulum, they have a very high protein lipid ratio. So they have protein so much more than lipids. Okay. So if you look at the protein lipid ratio, we have the myelin on one spectrum. On the other side, mitochondrial inner membrane, sarcoplasmic reticulum with abnormally high proteins. Okay. Now coming individually to each composition. We got your proteins. So when you talk about your proteins, there are essentially two types in your membrane. We have an integral protein, okay, and we have peripheral. Integral is also called transmembrane intrinsic, or peripheral or extrinsic is also there. Okay, so these can also come specifically as separate short notes. Describe about your proteins in the membrane, or describe lipids in the membrane. Okay, what are the functions of proteins in your membrane? So in various ways, questions can come like that. The one thing, we have transmembrane that runs completely throughout. Okay, and they are very integral. They have integral functions to this. So that is your transmembrane, integral or intrinsic. Okay, although the words are interchangeably used, it's not exactly that interchangeable. But at your level, I think it's okay to understand it like that. So what are some examples of integral or transmembrane proteins? So you can just think of all the things where you need transport across the membrane. Okay, so if you look at it, your pumps, what are pumps? They basically use energy, ATP is used, and pump something. So those kind of proteins are there. Okay, then we have carriers. So carriers are certain substances which needs to take something from one side and put it to other side. Like especially your facilitated transport, it may or may not require ATP. Carriers may or may not require ATP. Okay, but for some particles which are unable to cross through your lipid layer, you need certain structures like carriers. Okay, so that is the importance of all these proteins. You have the lipid bilayer. Things that are lipid soluble can easily pass through. Things that are water soluble cannot easily pass through. For that, you need specialized transporters, which are your protein structures. It can be a pump. It can be a carrier. Okay. It can be a channel where water per se can be transported. So it can be like, a complex like this, a channel through which now your water has to move in. It can be a pore complex. So just imagine there is a complex like this, a pore complex like this, through which your ions freely move. Okay, so all of these are essentially for transport. I hope you understand the difference between them. Okay, when I was a student at your age and I was studying all this, and all of them seemed same to me. I didn't know why it had to be separately mentioned as separate things. But all of them have quite a lot of different. That is why I'm trying to explain this to you. Okay, pumps, ATP dependent, carriers with ATP or without ATP, which are needed to, for example, facilitated transport. Channels for water movement. Pores, ions. Okay, and mostly these pores are not, do not require ATP. Simple diffusion is what happens. Now, apart from this, we have your receptors. Okay, so a lot of receptors are transmembrane in nature. And it is usually attached inside to certain other peripheral proteins. Okay, so there is one major receptor which is there like this, and this receptor runs throughout the membrane. So ligand comes and binds to it. So like that, your receptors are there. And apart from that, there are cell adhesion molecules. So for example, we hear of structures like cadherin. Yesterday I was talking about this. There's a structure called cadherin. When you talk about your desmosomes, okay, or adhering junction, when you talk about all of these, we have certain things like cadherin. These are transmembrane proteins which run across the membrane. Imagine there's another cell over here. They also have another transmembrane and they link to each other and connect to each other. So that is an adhering junction. Okay, so like that, these proteins are adhering, okay, adhesion molecules. And also certain components are there which are intracellular signaling agents. But mostly these signaling agents are your peripheral components. Okay, so these are examples. So under each of these heading, I want you, when you're writing an answer, to explain what they are, how they function, if possible, draw a diagram, and then give an example. That is how you get marks. So based on how much mark the question is. If, for example, the question is 3-mark question, and just the question is specifically what are the protein structures, then for 3 marks, obviously you have to write just this much is not enough. You have to write all of these things I've talked about. Okay, instead if it's just a part of a sub question of 1 mark, then these headings are more than enough. So you have to look at all of these things before writing. Okay, so that is your integral or transmembrane or intrinsic. Now, extrinsic proteins can either be partly there outside or it can be there inside. Can you see the difference? These are substances which are there outside. Okay, not completely going through the membrane, partially there. Or it can be present outside like this, or it can be present inside. So this is what I was talking about. Certain receptors have associated peripheral proteins which are there. Okay, so these can, once these receptors are stimulated, these peripheral proteins can act as a enzyme and act on something else. Okay. These can also take part in intracellular signaling. One common example is your G protein. What is the G protein? That is simply a peripheral protein with 3 different subunits. Okay, so this is an example of your intracellular signaling. Even enzymes, for that matter, can be part of intracellular signaling as well. Enzyme linked, not only your G protein type, but enzyme linked type of receptors are also there. Okay, now these can also act as cell adhesion molecules. For example, look at something like this, which is there peripherally, which can act as a cell adhesion molecule. Or again, if you look at something like cadherin, okay, usually cadherin is attached to something inside, like the catin, to which your actin comes and binds, or the intermediate filaments through plaques. Okay, so whatever it is, either the plaque or catenin, we have another peripheral protein inside. Okay, so that is an example of how this can be involved in a cell adhesion molecule. And then can be part of a submembranous cytoskeleton. What is a submembranous cytoskeleton? So again, this what I was talking about. We have a protein And attached to it, we have some actin, myosin, all of these cytoskeletal structures. So this can form one component, especially important in your RBC. You hear terms like spectrin and chirin. What are all these components? They are nothing but your peripheral proteins. Okay, so we have, even there, we have one major channel which is there, which is gantry, which is a transmembrane. Attached to that transmembrane inside, there is going to be your spectrin and chirin. And attached to that, we have your cytoskeleton. So this whole complex form a submembranous cytoskeletal structure. So understand? So if a question is asked about the protein components, you have all of these questions, and based on how much marks is carried, you can explain each of this with examples and diagrams. Okay, so this is one thing. Next is your lipid structures. Okay, so when you talk about lipid structures, we have the major one that we are talking about was phospholipid. So phospholipid, what does it mean? A phosphate group is there. Okay, majorly a phosphate with a glycerol, phosphoglycerin inside. That phosphoglycerin attached to fatty acid layers. Okay, so this is the structure of your lipid bilayer in your membrane. Okay, so we have fatty acids and on the other side, phosphate. And that phosphate group has a head of various components like ethanolamine, but it can also be serine, choline, inositol. Okay, so this is just an example of phosphatidylethanolamine, a type of phospholipid, but it can also contain, instead of ethanolamine, serine, choline, or inositol. You can draw this diagram. Sometimes a question like, with a neat label diagram, explain the structure of phospholipid and its important in cell membrane. This can come in biochemistry also, not only physiology. Okay, so that is one type of lipid, the phospholipid. Okay, then you talk about what is the importance of phospholipid. Phospholipid, what is the importance? It forms a bilayer, okay, because of which the water of the intracellular fluid and the water of the extracellular fluid are kept separate. Again, this lipid-soluble layer that makes sure that water-soluble substances cannot easily pass through, unless there is some sort of carrier or some protein structure in there. Only lipid-soluble substances can pass through. Okay, so selective permeability. And this membrane structure also helps in protection. Okay, so this is all the importance of this lipid structure, which is there. Now, apart from your phospholipids, we have certain other components. One example is your glycosphingolipid. Okay, one type of glycosphingolipid is your galactocerebrocyte, also ganglioside. So here, what is happening? Look at this phosphoglycerol part. You replace this phosphoglycerol part with a sphingosine component. That's why it's a sphingolipid. Okay, so just the same thing, the essential structure is the same, but there's a fatty acid layer. Instead of the phosphoglycerol, there is a sphingosine. Now, to that sphingosine, if you add a galactose, it forms a galactocerebrocyte, which is a type of glycosphingolipid, or in your nervous system. Another type is your sphingomyelin. Okay, here though, we have a phosphate with a choline head group. Okay, so we have the phosphate component, just like here, a phosphate with a head group. Okay, here, example is a choline. So that whole component is still same. But instead of the remaining part being exactly the same, we have a fatty acid component along with this, forming what you call a ceramide. Okay, so these are examples of lipids which are there. Apart from this, we also have your cholesterol. So these structures, again, phospholipid you can talk about. In the nervous system, you can talk about. Okay. And we have the cholesterol, which again has a polar head group and a tail part. Now, this determines the fluidity. Okay. So, for example, higher the cholesterol concentration, higher the fluidity. Okay. So, apart from cholesterol, even these lipids that we were talking about, phospholipids, their nature also is very important. So if more double bonds are there, or the length of the fatty acid chain is short, or outside factors like higher temperature, all of that will make it more fluid. So this is acting like a gel salt layer, can change based on the outside temperature and the components of the membrane. The fluidity can change. So this I seen as a separate short note. That is why I'm, going more into this. Factors affecting the fluidity of the membrane. So one is temperature. As the temperature changes, fluidity can be altered. If more double bonds are there in the fatty acid chains, or it is a short length of fatty acid, or another important component is your cholesterol. Higher the cholesterol which is there, more fluidity of the membrane. Okay, so when a question comes of what are the functions of your cell membrane, that can come as a long question. Okay, what are the functions of the cell membrane? I want you to think of each of these individual components and add these points also to your final answer. Okay, don't just be like, okay, they're just important for protection, selective permeability, and the few points which you commonly see in textbooks. But beyond that, cell membrane has so much more functionality. Okay, so one is proteins, then we have the lipids, and we have the carbohydrate components also. Can you see here these structures? These are your carbohydrate components. So these carbohydrate components outside, they help give a negative charge. Okay. They also act as, addition molecules. Okay, so they give sort of a negative charge, can act as cell addition molecules and all. Okay, so these are various components I want you to understand when you look at the composition and the functions of each of them in specific. Okay, so can you take a look at this? We have a fatty acid layer and a phosphoglycerol layer. Then we have sphingolipids, we have cholesterol. Okay, so sphingolipids again in the nervous system for signal transmission. Cholesterol maintains the fluidity of the membrane. Okay, so all of this I already explained with diagram. What is the composition of protein layer? Again, integral proteins are there, peripheral proteins are there. What are the integral proteins? Each of them, I had actually explained it to you also. We have peripheral proteins also. Yeah. So, the things that are there here are very minimal. So whatever I've explained to you, please note that down and write that for the exams. And this is the other question I was talking about. What are the functions of cell membrane? So you can talk about protection. You can talk about selective permeability. Okay. Part of this selective permeability is absorption of nutrients or excretion. Um, it helps maintain the shape and size. It helps your ICF and your ECF being kept separate. Okay. It is important for signal transmission, components of it. Okay. Uh, Tethering between the different cells, all of that components. Okay. So you can talk about all of these separate functions that I was talking about, proteins, lipid, all of that also can be included based on how much marks there is. Okay, next we'll go to something called apoptosis. So this, as I was telling you, recently it's been asked quite a lot. So you will have to know a little bit about it. Okay, so what is apoptosis? This is basically cell death, but by itself, a gene-driven activity. Okay, so this is gene-driven, requires ATP. Okay, ATP dependent, gene-driven. But the thing is, it does not completely cause a lot of havoc. It does not cause a lot of inflammation. Usually, whenever there's some sort of damage, there's going to be a layer of inflammation. Okay, cytokines will be released, inflammatory mediators will be released, and your uh, macrophages and all your WBCs will start coming there, causing a lot of havoc in that area. Okay, but something like that will not happen over here. That is the thing about apoptosis. You understand that, okay, there's some defect in that cell, or that cell it's about to die. Okay, so certain things, or it is filled with some virus. So how can we kill it off by a gene-driven mechanism? Okay, and there is no inflammation, and usually the membrane integrity is maintained. Okay, only the later stages it starts fragmenting. So through the whole process, it starts, changes happen inside without damage to the membrane, no inflammation. Okay, and slowly shrinks, and then it gets damaged. But what are the components of it or the functions? That is very important. Okay. Uh, okay, so I'm gonna switch over. Again, this much is not at all enough for a question. You'll hardly, if a question of 5 marks or 3 marks come and you write only this much, you will hardly get 1 mark maximum. Okay, so again, apoptosis, as I was telling you, there is gene-driven mechanism, requires ATP. Okay, membrane integrity is maintained, and there is no inflammation. Okay, this is as opposed to something else called necroptosis, where membrane integrity is not, the inflammation is there and all that. Now, when you talk about your apoptosis, the steps in apoptosis, this is the simplified version of this. If you have to study the day before exam and you have no idea, then this is most basic way I can teach you. So for apoptosis to happen, the signal can either come from outside, okay, or it can come somewhere from inside. So some intrinsic signal can be there, or something from outside, an extrinsic signal can be there. So we have certain ligands, fast ligand, death receptors, different components like that are there. So when ligands bind to this from outside, that will activate. So caspases are certain protein structures which actually, like, they're basically enzymes. Okay, we have initiator caspase, executioner caspase. So a lot of initiator caspase keeps getting activated. Ultimately, we have the main executioner caspase. Once that is activated, your apoptosis is going to start. Okay, so your death receptor, one is extrinsic signal from outside, will activate what you call caspase 8, a type of initiator caspase. Okay, other than that, your intrinsic signals can activate caspase 9, for example. But ultimately, all of them will go and activate your executioner caspase, which will start. So again, you have caspase. For caspase, you have extrinsic and intrinsic. Extrinsic, like for example, death receptor mediated, caspase 8 is activated. On the other hand, intrinsic, some signal from inside, either it will be in your nucleus or in your mitochondria. But ultimately, that is going to go and activate caspase 9, and that will activate your executioner caspase. Okay, so this nucleus or the mitochondria, what happens if there is some sort of a nuclear damage which is detected? Okay, or some sort of a mitochondrial damage, or sometimes there's nuclear damage by itself, through certain genome structures and all, can go and activate the mitochondria to help release caspase 9. Okay, so there is a transporter called MPTP. So that gets activated. So ultimately, when you look at your gene, we have something called the BAX and the BCL. Okay, so we have a pro, which is for this, and we have an anti. So if BCL is there in high number, that is anti. It prevents this. But if BAX is there more, it results in this activity. So based on the, how much amount of this is there under various conditions. Okay, this is all mediated by some P53 components and all. All of this you learn later. So ultimately, what you have to understand, there's a nuclear damage or a mitochondrial damage, which will result in caspase 9 activation. Okay, ultimately, executioner caspase is activated. So this is how apoptosis acts. So first thing you have to talk about apoptosis. How does apoptosis work? What are the components and what makes it different from other kinds of cell death? Okay, two, what actually happens here? Okay, so you'll have to talk about this. Next thing, what are the functions? So one example is in the embryogenesis. So in embryogenesis, what happens is, like, take a look at your limbs, for example, your digits. Okay, the digits that form comes out as a lump like this. And then what happens? Through apoptosis, this part dies out. And you'll eventually get, once it goes into apoptosis, you'll eventually get your fingers and all. So in various stages of embryogenesis, apoptosis is used. Okay. Apart from that, male, female. At birth, we have both male and female structures, the primitive structures. Once you are a male, then the female structures regress. So such a regression of the Mullerian structures also is through your apoptosis. Now, this is something we learned called immune tolerance in immunity. So the cells which are formed, your T cells, for example, those T cells go into thymus, lymphoid organs and all. So over there, clonal selection takes place. Okay, it checks to see if they are reactive against self tissues. If certain group of your T cells react against your own self, then through apoptosis, the thymus will make it go through negative selection and remove it. So for immune selection, this is important. Okay, so under each of this, I want you to explain all of this, not just write the headings. Okay, then menstrual cycle. So menstrual cycle, if you look at it, there's no inflammation per se. Okay, so what is happening is the sloughing off that is happening over that time. There is a lot of apoptosis which is happening. And destroy dangerous cell. So either it's a virus infected cell, okay, or some prion structures are there, misfolded proteins. Okay, so all of these changes that are there in a cell, that cell gets killed off by itself, suicide. Okay, so these are various functions of your apoptosis. So under apoptosis, you have to talk about what is apoptosis, how does it act, and what are the functions of apoptosis. Okay. Now we'll come to the transport, but before that, there's one small part of your general physiology, which oftentimes questions come from. Okay, so that is why I want to highlight this particular part. So this is very basic. It's about, the extracellular fluid, intracellular fluid, body composition, small components of that. Okay, so a 60 kg man, if you take a look, 70 kg man, if you take a look at it, 60% of the body weight is going to be water. That comes to around 42 liters. I want you to remember these values because they come often in MCQs, fill in the blanks and all that. Okay, this by itself can come as a question, a short 1 mark question, 2 mark question where you have to explain all of this. So 60%, that is 42 liters water. 2/3 of that is ICF. Out of all the water which is there, 2/3 is in the ICF. That comes to around 28 liters. Whereas 1/3 of it is your ECF, and that comes to around 14 liters. Okay, so if you're saying 60% of the body weight, okay, so 40% of your body weight is ICF, 20% of your body weight is ECF. So don't get confused. This percentage is percentage of the body weight. Okay. Percentage of the water means this is the thing you have to convert into percentage. So be wary of what the question is. People will simply try to confuse you for the exams. Then out of the 14 liter, which is there, 11 liter is your interstitial fluid and 3 liter is your plasma. Okay, so for all this, it'd be wise to even draw a diagram. So for the exam, I always want students to draw diagrams and explain. It makes it much more easier for the teachers who are correcting, because most of the time teachers don't have time to, sit and, look at everything and correct. So it'll be very helpful and not piss off teachers who are correcting if a lot of diagrams are there. You write it in point formats. So they just scan through and they'll easily give you marks. So we have your ICF over here. This whole is your ECF, and in your ECF we have the interstitial fluid, and this is the plasma, and these are the various percentage of it. Now sometimes what are the substances that can be used in order to measure them can be asked as a question. Okay, in which case, if you have to measure total body water, you can use heavy water, deuterium oxide, radioactive water, tritium oxide, or aminopyrine antipyrine. Okay, if you use substances for plasma measurement, you can use Evans blue, radioactive fibrinogen, radioactive albumin. For extracellular fluid, you can use sodium thiosulfate or sucrose mannitol inulin. Okay. Now we'll come to your transport. So when you talk about transport, we have these various headings under which. So this, if it comes as an essay, a long question, you have to talk about all of this. You have to understand the outline of what are the headings you have to cover under transport. One, we have your passive transport mechanisms. We have your active transport. Okay. Now, beyond that, we have to talk about vesicular transport later, where you can talk about endocytotic mechanisms, exocytosis, and all that. Okay, which we'll come to later. But it depends on how much mark there is. So usually students, when an essay of transport mechanism come, they only focus on this and this, and end up forgetting about this part, which is also equally important. Sometimes separately a question on this part can come. Okay, so you'll have to know each of this heading properly. So what is simple diffusion? You take a membrane, okay, and let's take a pore, for example, which I was talking about, transmembrane structure, a pore. You put a bunch of ions over here, which are more over here and less over here. By Brownian movement, obviously, this is going to go in this direction. That is just simple diffusion. Okay, based on the concentration gradient, this is moving. Now, to determine how much amount of the substance is going, for that, we use this Fick's law. Okay, so under Fick's law, you have to understand one thing we already covered here. how much was the concentration gradient. If more was there this side and less here, it'll move towards this side. So the rate of flow, the flow rate is directly proportional to one, the concentration gradient. Now imagine the thickness of this is higher. Okay, it's this thick. Would it be easier to cross this or more difficult? Becomes more difficult to cross it. Okay, so thickness is going to be something which is inversely proportional. And what about the surface area? If there's a pore like this, versus a pore like this, where would the flow be faster? Obviously, it's going to be this pore. Okay, so again, surface area is also something that is directly proportional. So this is ultimately a fixed law. So what determines how fast the simple diffusion is happening, the flow rate is happening? One, the concentration gradient between them. Two, what is the surface area? Three, what is the thickness? Okay, so again, going back to this, you can understand Fick's law, A delta C. Okay, so over here, this proportionality, if you were to change it and put a constant, that is your D over here. And apart from this, actually, for Fick's law, I'd want you to use your thickness also. Okay, not only your area, thickness is also important. Okay, so again, I already explained to you the Fick's law. That is to understand about the Brownian movement and simple diffusion. Now, what is simple diffusion again? Simple diffusion either can happen in ungated channels by itself, like the one I was showing you. Okay, ions can pass through, or it can be something lipid soluble, which can easily. So lipid soluble substances can easily pass through by simple diffusion. Or there is a water soluble structure which requires some sort of channels or pores, okay, for movement. Now another question, can you see this? We have a simple diffusion. This is the ungated channel, or a channel like this. Okay, for any reason it gets stimulated, then simple diffusion can happen. Otherwise, it remains closed. So these are all examples of simple diffusion. One question that can usually come are, what are the factors affecting simple diffusion? Okay, which you can think about the Fick's law only. What are the factors affecting one concentration gradient? Similar to concentration gradient, pressure gradient is also involved there in certain aspects. Then we have the area, we have the thickness. Then apart from that, what is the solubility? Like, is it water soluble? Is it lipid soluble? Okay, so all of these are factors that affect your flow rate. Okay. Next is facilitated diffusion. So what is facilitated diffusion? Can you see here? This carrier protein comes, binds to it, and then it is taken this side. Okay, so either it's take before this, what is happening is this usually is there like this. Okay, and whatever molecule is here goes here. And then conformational changes happen where it now opens to this side and goes over here. Okay, so you understand this. So this is basically your facilitated diffusion. So compared to your simple diffusion, this is not something that can easily happen. It requires some protein, a protein like this, in order to take it and put it to the other side. Okay. So because a protein is required like this is where I want you to draw a diagram also. Usually a question that comes is, what is the difference between a facilitated diffusion and a simple diffusion? A question like that often comes. Facilitated diffusion is a simple diffusion. So in facilitated diffusion and simple diffusion, what is the same thing? They're both passive. They're both passive, and the movement is based on concentration gradient. And both do not require ATP because it's moving based on concentration gradient. So these are basic aspects about this. Now, how is it different in facilitated diffusion? Saturation kinetics is present. The main reason for that is there is a need for a carrier protein. So what does it mean? If you look at the rate of flow, okay, with change in the concentration gradient. With increase in the concentration gradient, the rate of flow increases, and at some point will get saturated. On the other hand, if you look at your simple diffusion, as the concentration gradient increases, it keeps on increasing. So this constancy is because of the presence of the carrier protein. But even if the concentration gradient increases, the carrier protein number is not increasing. At some point, all the carrier proteins, maximum it can do is reached. Okay, so you draw this and explain this concept also. So this is why there is a saturation kinetics over here. Okay, so that is the difference between your simple and facilitated diffusion. One example for facilitated diffusion is your GLUT transporters. Okay, your glucose transport, which is happening. This is an example of a facilitated diffusion. Okay, now one thing that is not given here is a third type, which is non-ionic diffusion. So when you talk about your passive, so this is if it comes as a long question, you'll have to talk about simple diffusion, talk about Fick's law, okay, factors affecting simple diffusion. Then you talk about facilitated diffusion, what is the difference between simple and facilitated diffusion. Then you go to non-ionic diffusion. What is non-ionic diffusion? So certain components, your H2CO3, for example. Okay, this H2CO3 becomes H plus and HCO3 minus. Okay, and H plus and HCO3 minus can combine. So this is a reversible reaction. Now these ions cannot easily pass through a membrane. It needs transporters. But there is a way to bypass this. This H2CO3 can instead become CO2 and water. Okay, and this is something which can easily go lipid soluble, where it can now combine with water inside and go and reform H2CO3, which can now give H plus and HCO3 minus. Okay, so over here, was any transporter required? No. Was ATP required? No. This was a sort of a diffusion where the ions are converted to a non-ionic form and then transported inside. So this is an example of non-ionic diffusion. Something you very commonly see in physiology, especially your renal physiology, for example. Okay. Now beyond that, we have your active transport. Active transport is anything that requires energy, either directly or indirectly. The primary active are examples where direct ATP use is there by the transporter. Or in secondary, these transporters do not use ATP. Okay, there is some associated primary active transport which is already happening. As a result of that, a secondary active transporter can now do its activity. So although it does not directly result in any sort of ATP usage, indirectly it is involved. Okay, so active transport Again, why is ATP required here? Because this is moving against a concentration gradient. So in facilitated diffusion and simple diffusion, what did we understand? It is going according to concentration gradient. But in active transport, what is happening? It is moving against your concentration gradient. Okay, hence it's called uphill. And thus it requires energy, because to move against concentration gradient, you require energy. Okay, that energy usually from the breakdown of ATP. Okay, this I'll talk about it a little later. So we have primary active transport. All of this we already talked about it. What is passive transport? Okay. So in primary active transport, the main question that is going to come is sodium potassium ATPase. Apart from sodium potassium ATPase, we have other examples like in your stomach, acid secretion. Okay. Or your calcium ATPase, sarcoplasmic endoplasmic reticulum, calcium ATPase. Okay. Active transport of calcium into your endoplasmic reticulum, sarcoplasmic reticulum. So that is one thing that can happen. Then we have the hydrogen potassium ATPase, which is happening for acid secretion, or sodium potassium ATPase. These are examples of primary active transport. In that, your sodium potassium ATPase is pretty important, which can come as a long question or can come as a part of a sub question of an essay. So here are a few things. I want you to draw a diagram like this. The sodium potassium ATPase has a alpha and a beta subunit. Okay, now most of these things happen in your alpha subunit. In your beta subunit, there's some glycosylation unit. Okay, apart from that, your alpha subunit has various components in it. It has one area where sodium will bind. It has one area on the outside where potassium can bind. Okay, there's one area where ATPase activity is there. So what happens? Your ATP gets broken down to ADP and inorganic phosphate. Now, this inorganic phosphate specifically goes to one particular area, that is aspartate 376 position of your this amino acid sequence. It goes and binds there. That is how ATP is utilized. And then a conformational change is happening. So in its resting state, your sodium from inside is gonna come and bind over here. Okay. And from the outside, potassium is gonna come and bind over here. Okay. That will result in a conformational change, and potassium will go inside, sodium will go outside. This is again very basic version of the whole thing. It's a little more complicated than this, but at your level, this basic description is more than enough. Okay, you just have to know alpha subunit is there, beta subunit is there. It has ATPase where ATP will be broken down and will go to aspartate, and that will result in a conformational change. Okay, there is also one small part where a particular substance can come bind on it, obey. Okay, for that also there is a small part over here. So sodium binding, potassium binding, it basically flips. Okay, so 3 sodium and 2 potassium. Okay, so this is important because this is what we call electrogenic when we talk about the functions of sodium potassium ATP. So first draw the diagram, explain how it works and all that. Then you talk about the functions. The first function is it is an electrogenic pump. Electrogenic, why? Because 3 sodium is going out, 2 potassium is coming in. So ultimately, because of that, what is happening? The inside a negativity is developing. Okay, so each time a 3 sodium goes out, 2 potassium is coming in. That means we are having one deficient. inside. Okay, that helps maintain your RMP. It helps in maintenance of your RMP. Okay, because your RMP is usually minus 90 millivolt. So such a negativity is maintained by your sodium potassium ATPase. Otherwise, if positive ions keep leaking inside, like sodium, for example, it'll come more towards the threshold. So to avoid that, we have a mechanism like this. Okay. Now, another thing is, if, for example, a lot of positively charged ions come in, that also by osmosis will fill up the, increase the volume. Okay. And it'll ultimately result in rupture. So to prevent something like that, again, this helps maintain volume. Okay. If you look at your body, majority of your energy utilization, ATP, is utilized for your sodium potassium ATPs. This is very important in your body. Not only does it, act to maintain your RMP or maintain your volume, but it also creates a gradient for secondary active transport. This is what I was talking about earlier. For secondary active transport to work, some degree of primary active transport should be usually present. So, for example, what is happening? Sodium is being taken outside. So now inside the cell, sodium is going to be slightly deficient, which means now another transporter can be kept here, where now sodium can come inside. Okay, so although this was just a standard diffusion, which is happening, this happened because of another primary active transporter. Otherwise, it would never come in like that. Okay, so this is an example of a secondary active transport. And usually when this happens, along with that, some other movement also is usually there. For example, the sodium comes in. Okay. In exchange to that, another ion usually goes out, a positively charged ion. So that is what you call antiporter or exchanger. Okay. Instead, one thing comes here. Okay. Along with that, another molecule also comes inside. Then it is called a symporter or co-transporter. Okay, so these are how you can describe a secondary active transport also. So coming back to our sodium potassium ATPase. So these are the various functions I want you to talk about. Then next you can talk about what are the factors that affect sodium potassium ATPase. So whenever there is thyroid hormone increases your basal metabolic rate in the body. Thyroid hormone increases the pump activity. Aldosterone also increases the sodium potassium ATPase activity, thereby creating the gradient for sodium reabsorption to take place actually, and potassium excretion. Okay, you'll learn about this in your renal system. And then insulin also to a degree affects the sodium potassium ATPase. On the other hand, this is a common question that can come. True or false, or give a reason why. So in that, digitalis, how does it act? It acts by decreasing the activity of sodium potassium ATPase. So this whole mechanism that we just talked about, what does digitalis do? I'm just going to describe it because it's a very important question. So associated with your sodium potassium ATPase, what does the sodium potassium ATPase do? Sodium outside and potassium inside. That just created a gradient for sodium now to come in. And in exchange for that sodium, a calcium is going outside. So if, for example, in heart failure, we stop the sodium potassium ATPase, there will no longer be a gradient for sodium, which means calcium won't go out, which means calcium will remain inside, which means more calcium inside means contraction will be plausible. So this is what we target in heart failure by using digitalis. For that, more calcium is available for contraction to take place. Okay, so here is a classical example of your primary active transport and an associated secondary active transport. And what type of secondary active was that? An exchanger or antipode. So I hope you understood the meaning of all these words. Okay, now beyond this, hypoxia, hypothermia, all that also decreases the pump activity, and certain metabolic poisons like 2,4-dinitrophenol can also inhibit the pump activity. Okay, so that is a very important question that very often comes. Primary active transport, in specific, your sodium potassium ATPase. Okay, secondary active transport also I in detail explained, and I've given examples. Counter transport, one example is sodium calcium exchange. Okay, there are also sodium hydrogen exchanges. In co-transport, one example is your sodium potassium 2-chloride transporters, which are there. Okay, or for example, sodium and your glucose coming inside, or your sodium and amino acid, or sodium iodide. All of these examples of co-transporters, they go in the same direction. Okay, so you have to, when you write these answers, for each of this, you have to give examples also, very important. Marks are there for that. Okay, sodium potassium ATPase, again, I already explained the mechanism. Okay, you can draw a better diagram than this, the one that I showed you. Okay. Yeah. So next, coming to what is phagocytosis. So we talked about these headings: passive, active, simple diffusion, facilitated, non-ionic, primary active, secondary active. So this again can come as a long question. Now, apart from that, we got what is phagocytosis. Okay. So phagocytosis, usually it is for substances which are less than 0.5 micrometer in diameter. Okay, not less, I'm sorry, more than 0.5 micrometer. So you have to understand, when you learn this, you can pick up a 0.1, 0.2 to 0.5, and more than 0.5. Under that, you can study various headings. Less than 0.1, 0.2 to 0.5, more than 0.5. So more than 0.5, you will use phagocytosis. Okay. If it is less than 1, you can use pinocytosis, micropinocytosis. And you can use macropinocytosis if it is 0.2 to 0.5. Okay. So we have pinocytosis and phagocytosis. What is the essential difference between phagocytosis and pinocytosis? So in phagocytosis, whatever component is there, once you engulf it and take it in. Okay, so in phagocytosis, there is a structure like this. Okay, now something is coming from outside and it is detected. Okay, this receptor also, it gets detected. Now, once it gets detected, so that is the first thing that happens, detection. Then pseudopod actin polymerization and all happens, pseudopod formation happens, and it gets internalized, resulting in a substance called phagosome being formed. So inside this, once it is internalized, okay, there is a area which is formed with that substance which came from outside. Now this substance is drained clear and shrunk properly till basically only this structure is there, surrounded by a vesicle. Okay, so that means whatever things are there outside this, like the fluid, is not taken in phagocytosis. Okay, now there are various structures here called endosome. In some way, this binds to endosomes, buds off, a lot of processes like that happen. And ultimately, once this goes into the endosome, now lysozymes which are here will come and degranulate into this to result in hydrolytic enzymes to be released and degraded. So these are steps in phagocytosis. Again, what is it? 0.5 micrometer, more than that. What are the steps in phagocytosis? 1, detection. 2, internalization. 3, phagosome formation. 4, phagolysosome. Okay, and you can draw the steps and explain also. So this is your phagocytosis. Okay. Okay, so can you see here? There's a receptor, it binds, it comes here, and then it forms a phagosome. And from this lysosome also goes to this, and it is degenerated. Okay. Now, other forms of endocytosis which are there, we have pinocytosis and receptor-mediated endocytosis. Okay, so we were talking about phagocytosis, which is one type. Apart from that, we have pinocytosis. In pinocytosis, what is going to happen is a cell, okay, and a structure is coming, and like this actin polymerization happens, and this whole component closes, and you have ultimately a vesicle filled with whatever was there outside also. Okay, so even the fluid is coming in, not like phagocytosis. This is an example of your macropinocytosis, fluid drinking. And apart from that, very small substances can easily come in along with the fluid it is encapsulating in. This is what we call micropinocytosis. This is what we call fluid phase endocytosis. Okay, for that, we need some clathrin, caveolin, all of these components are required. Okay, so pinocytosis is cell drinking, which can be divided into macro as well as micro, depends on the size and how it is happening also. Then we have non-phagocytotic. I just talk about two things, clathrin and caveolin. What is clathrin? What is caveolin? What is the difference between them? Just look at this diagram. Okay, so these are what are called receptor-mediated endocytosis. So here what happens is, one, can you see a receptor here? Okay, so this ligand comes bind to it. When it comes and binds to it, there is a particular substance called clathrin. This clathrin has a triskelion structure. It has a structure like this, like 1, 2, 3. Okay, where it has a particular component called adaptin. Now, once this comes, this will go and attach itself to the receptor. Okay, so what happens is this membrane actually starts budding like this. And with it, more and more of these triskelion keep attaching, multiple ones, forming a whole lattice network over here like this. Until finally, a GTPase will come, cut it off, resulting in a complete vesicle forming with a lattice network of your receptor and your clathrin. Now, ultimately, this will go to your endosome. Once inside the endosome again, lysozymes will degranulate into this. And that will help separate your receptors and your triskelion, which will now go back. But whatever molecule was taken in, that has come in. So this is one type of mechanism where a clathrin structure is involved, which is the triskelion, adaptin, okay, and it comes in. Now, apart from this, there is another mechanism where there is a GPI. Yesterday we were talking about it. Okay, a GPI linked receptor is there. Okay, so usually in the membrane, there is a lipid rich layer. Lipid rafts, we call it. This lipid rich layer usually has some substances like caveolin. Okay, so lipid rich layer, caveolin. Now, in a similar fashion, when something binds to it, this whole thing again comes in shaped like this. Multiple caveolin layers are there, and that also through GTPase will bud off. Instead of endosome, this will go to another structure called caveosome. So that is the essential difference between what a clathrin is, what a caveolin is. Okay, and these are examples of receptor-mediated endocytosis. This is just another diagram for looking at this. Can you see? This is a receptor ligand molecule. This is an adapted. This is a triskelion structure. And this vesicle, it is coated. And you can see here again how this can go endosome, and ultimately your lysosome also can bind to it. Apart from that, over here we have the whole caveolin structure also. Okay, so clathrin mediated endocytosis, caveolin mediated endocytosis. So this is what you have to talk about in endocytosis: pinocytosis, phagocytosis, receptor mediated endocytosis. Okay, in pinocytosis, we talked about cell drinking. Okay, receptor mediated endocytosis, again, I explained in detail. Exocytosis, again, a process by which things are put outside, expelled outside. So for that, we have your V snare. Usually a vesicle is there. Okay. That vesicle has a V snare and a T snare, which is there on the membrane, and both of them combine. And there's a whole lot of processes of exocytosis. Usually calcium is involved. Whenever calcium comes in, something called synaptogmin is there. Calcium binds to it, and then it results in a conformational change. And ultimately, a fusion synaptophysin is there, and a fusion takes place, and exocytosis takes place. So these are various steps that happen in exocytosis. So we have V snare and a T snare, and they will come dock against each other. Okay, for this your calcium which is there, that will come and bind to synaptogmin, and that will result in your change in the structure of this V snare T snare, make it come closer, and a synaptophysin will make it fuse, and exocytosis will happen. Okay. So ultimately, this is what we have covered. What are the functions of cell? What is apoptosis? What is Fick's law? Different types of transport, that is simple diffusion, facilitated, active, passive. Again, what are your primary active and secondary active transport? Also, sodium potassium ATPase more in detail. Okay, and apart from that, your endocytosis, exocytosis mechanism, examples, what is phagocytosis, all of those questions also I covered. Okay, so these are the various things that we have to talk about. So thanks for watching. If you want to get access to more content like this, you can go ahead and follow Auricle's social media handles in your YouTube, Instagram, Facebook, as well as your Telegram, where you can get notes and you can download the app. It's available in Play Store. So thanks for watching. See you again soon. I think we can end the session now.